cycle. We stopped here, the last lecture. So we talk about glycolysis, we talk about Krebs cycle, right? And then in the Krebs cycle, there are three components: the citric acid cycle itself, the electron transport chain, and oxidative phosphorylation. We had time only to cover electron transport chain, but have not gone into oxidative phosphorylation, which is the production of ATP. Why do we have such processes going on, right? Why do we need to have three different types of processes? If we look at the issue lies with the structure of the mitochondria, right? We said that the mitochondria is quite special. This is the energy powerhouse, yet there are serious limitations of substances going in and out of the mitochondria. Right, so far our glycolysis processes, they are occurring outside the mitochondria in the cytosol, right? And we don't harvest enough ATP from the glucose. Glucose has 6 carbon, but at the end of glycolysis we only get 2 molecules of pyruvate. Right, so for complete oxidation, we need to break it down all the way to carbon dioxide and water. Completely break down the whole structure, whole backbone. But the first step, glycolysis, can only do it under 3 carbon. So in order to continue this complete oxidation, we need to bring the materials into the mitochondria, okay, to continue. So if you look at the matrix, the outer membrane of mitochondria is freely permeable to small molecules and ions. Inner membrane is impermeable to most small molecules, including protons. Alright, and then the matrix itself is where all the important enzymes are. The pyruvate dehydrogenase complex to make our acetyl-CoA, citric acid. Now today we're going to look at fatty acid, beta oxidation enzymes, and then next week amino acid oxidation enzymes, etc. So everything is compartmentalized in this mitochondria. It cannot come out. So materials have to go in. Okay, materials have to go in for reaction. So in order to facilitate this transport inside, we have quite a number of what we call transporters. These are proteins that act as pores or channels, open and close, okay? Like gates to allow these materials to go in. And we saw during the last lecture all this whole respiratory electron carrier. It is a complex, multi-enzyme complex of complexes 1 to 4, right? And their major role is to pass the parcel, right? What is the parcel? Electrons, right? Electrons from all the processes. Okay, then let us look at the picture here. See, this is the outer membrane, this is the inner membrane. Okay, so you have all these 4 complexes, right? Right, the electron transport chain, you have many of them distributed along the inner mitochondrial membrane. And so they themselves undergo a series of oxidation or reduction to pass the electrons. Right? So now the pyruvate is transported from outside cytosol into the matrix, okay, for acetyl-CoA production. Acetyl-CoA then undergoes citric acid cycle, everything inside the mitochondria. And during the citric acid cycle, we have production of 3 NADH and 1 FADH2. So this NADH is reduced, and we need to regenerate it to continue the citric acid cycle process. So there must be a way, right, over here, okay, NADH has a pair of electrons. Okay, it has taken up a pair of electrons from the pyruvate, from the acetyl-CoA and the proton. So now the pair of electrons have to be passed on to these complexes, right, in a sequential fashion, and finally to complex 4. Okay, complex 4, which will then pass on to final electron acceptor, which is the oxygen, to be reduced to water. Right, this is our end point. Right, only here then did we see oxygen's role in this. Right? Throughout the whole process, we cannot use oxygen as an oxidizing agent because it's too reactive and it produces radicals that are harmful to the cells. So at the end, then we use it to change it to water. Right? Only truly in this sense, then the electrons have been removed. Okay? So during this whole process, we also talk about a proton motive force because the electrons are associated. Every time you pass two electrons, you also have two protons, right? A pair of electrons, you also have protons being produced. And these protons, we know, concentration protons or protons will affect the pH of the matrix inside here, right? But in order to maintain all our functions, the pH has to be physiological, okay? Have to be pH 7. So there must be a way to remove it, right? Remove it and then pump it back again. So for one cycle, it has been found that 10 protons are removed. I mean, for one molecule of oxygen that's been reduced, we have 10 protons that are being pumped out into intermembrane space. But it doesn't stay there, it cannot stay there. So if it stays there, what happens? You will have accumulation of protons here, you will be relatively negative over here. Not only do you have a chemical potential due to the concentration of protons, you also have an electrical potential across the membranes, right? This is highly destabilizing. Or if it doesn't get pumped back, we say that the potential becomes an energy barrier. It becomes so hard, okay? The energy becomes so high that this reaction will not be able to overcome this high energy potential, and the reaction, the electron transport chain, will completely stop. And when electron transport chain completely stops, that's the end. That's the end of the mitochondria, and that's the end of us. Okay? So there must be a way, okay, to pump it back. So they have done experiments to find out that, well, before that they have already noticed that there is a particular protein complex and enzyme, okay, protein complex that has enzyme function called ATP synthase on the inner membrane. And this synthase is responsible for catalyzing, okay, the formation of ATP from ADP and inorganic phosphate. But what they subsequently found was that if they poison or inhibit this ATP synthase, they realized that the electron transport doesn't function. Okay? Consequently, if they poison this, you can easily poison this electron transport by cyanide. Okay, that's why you die, right? People get cyanide poisoning. What does it do? Cyanide inhibits these complexes. Okay? That's why you don't have energy production after that, and then you die quite quickly. Right? So cyanide over here. You can use cyanide to poison this electron transport chain. When you poison this, you don't have ATP synthesis. Conversely, if you poison this using other inhibitors, this one also doesn't work. So that's how they find out that actually these two processes are coupled. Right? Before that, the knowledge is not sufficient for us to know that. But after that, they notice, okay, it's actually coupled. One does not happen without the other. So how does this coupling works? Okay? How does this coupling works? Right. This coupling works because of this oligomer, complex oligomer that I've already mentioned called ATP synthase. Right? I'm going to let you watch a video first, and then I'll explain it, and then we will watch the video again to reinforce the whole idea. Okay, but just look at this structure here. Okay, the video you see over here, you see this flower-like structure. Okay, this is on the end side. This is on the matrix side. Okay, and this is a barrel and with a shaft. Okay, it's like a rotor, right? With a shaft that rotates, a pore here that rotates here, yeah. And this is on the intermembrane side. Okay. So during the picture, you will see all these. During the video, you will see all these elements. Yeah.
ATP synthase is a molecular machine that works like a turbine to convert the energy into chemical energy stored in the bond energy of ATP.
Upside down, this is the shaft.
The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. To show the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
Okay, so what is happening? Let us describe in detail. You saw this, right? This is, it was like kind of inverted on the intermembrane side. You saw those little square cubes, right, moving. Those are protons, right? And this is on the intermembrane space. So the protons were moving, and then the movement of the protons back into the matrix causes this barrel to spin around, right? You saw that. And once it spins around, it kind of rotates this shaft called the gamma subunit over here, the green color here, right? And the gamma subunit is in very close proximity of this three pairs of subunit called alpha beta subunit. Now, this doesn't move. This flower-like lollipop structure doesn't move. Instead, it changes conformation. Okay, the shaft is the one that is moving, but this one doesn't move. Okay, and once it changes conformation, it can catalyze the production of ATP. So how does it do that? Okay, so this is the F0, F1 synthase. Okay, it's called F0, F1 because this is the F1 synthase, this is the F0 subunit. So F0, F1, ATPase. The ATPase is the enzyme activity, okay, here, right? So now, okay, so we look at the subunit in a clearer picture, okay? At any one time, each of the subunit will adopt a different conformation, right? So this green arrow is the shaft, right? The green arrow is the shaft that is rotating, and in very close proximity. So when the green arrow, so you at any one time you will always have an open site, okay? Open site has a low affinity for the substrate. That means it has kind of just expelled the ATP that is being produced. right? Then you have a loose binding site, okay, which binds the substrate ADP and PI. And of course, the tight binding site, which is catalytically active, right, and it binds the product, right? It converted the ADP and PI to the product ATP over here. So when the protons get pumped, right, from the intermembrane space back into the matrix, the shaft will move. And the shaft moves in such a way to get into contact the subunit that has the ATP being produced, right? And then the ATP, the subunit with the ATP produced will then change its conformation. It doesn't move, right? It just changes its shape to expel the ATP, right? And this step doesn't happen. Okay, this step doesn't happen if you don't have the substrate being attached in the adjacent pair of alpha beta subunit. So it's a highly coordinated machine in this case, right? Imagine this production line. I've gone to see this Yakult factory, looking at the little empty bottle Yakult, being fed through the machine, filling, and then being sealed with the foil. So you can imagine this is like a production chain, right? You have the ATP already being produced, but next to it you have substrate bound. And once this occurs, the movement of the shaft will kick the ATP out. Now, once the ATP is kicked out, this becomes an open site. And the next site, which was previously occupied by the substrate, changes its conformation to facilitate the enzyme reaction to make ATP. Okay? That's why you see the raw products now is being changed to ATP. Right? And once this is done, again, another move or rotation of the shaft, this ATP that is being produced will be kicked out, okay, back into the matrix. So this is how the F0, F1 synthase works to produce ATP. Okay, so the energy in the electrons, although the electrons are being passed on finally to oxygen, but there is still a lot of energy left, right, from the protons that are moving across the membrane. And then this energy is then being interconverted to produce ATP. ATP is the chemical energy that we can use practically for our cell functions, right? So nothing is lost. It goes back to the first law of thermodynamics. Nothing is lost. It's just being interconverted to different character. Okay, to different character. All right. Okay. So you understand? So now, do you want to watch it again, or I can send you the link? You can go back and take your time to watch it. All right, I will send you the link. Yeah. Yeah. Okay. So that's the how ATP is finally being produced, and that's the end. That's the end already. All right. Okay. That's the end. So, but we have not addressed the other issue. Okay. The other issue is during glycolysis. Did you remember that? In glycolysis, we have a step, okay, that produces NADH, right? And we said that this NADH, if you were to do a sprint, this NADH is regenerated by lactate formation. That is a short-term kind of regeneration of NADH, right? Okay, so with a short-term kind of NADH is not sufficient. Eventually, it's better if this NADH produced from glycolysis can enter to the mitochondria to work. But the problem is, again, mitochondrial membrane does not allow many things to go through, so the NADH doesn't cross. Right? So in order to transport this NADH or the electrons into the mitochondria, we have what we call a shuttle system. Okay? And this shuttle system transports metabolites between mitochondria and cytosol. We have two. The first one is called glycerol phosphate shuttle, okay, which is in the skeletal muscle and brain. Right? It's found that for each cytosolic NADH, so this is from glycolysis, 1.5 ATP is being produced in the mitochondria. And if the malic and aspartate shuttle is being used, which is found in the kidney, liver, and heart, for each cytosolic NADH that is being transported, 2.5 mitochondrial ATP is being produced. Okay? So we'll look at how this shuttle works, okay? And then you will realize that what is really moving is the electrons, it's not the NADH. Okay? So NADH is not moving into the mitochondria. Let's take a look, yeah? So remember this step. Okay, this step is cytosolic. Okay, this is glycolysis, right? Okay, this is glycolysis process. Yeah. So remember the glycolysis process, we said that there is NADH being produced, right? NADH produced. And NADH being produced needs to be oxidized back to NAD so that glycolysis can continue. Okay? So what does it do? In this case, for the glycerol 3-phosphate shuttle, okay, there are two isozymes, okay, two isoforms of this enzyme called glycerol 3-phosphate dehydrogenase, which we have already seen. One of the after the fifth step, remember that we have 2 3-carbon compounds that are being produced. Glycerol 3-phosphate, which is GAP, and dihydroxyacetone phosphate, and they can, all of them will be converted to glycerol 3-phosphate to continue the glycolysis process. And this is catalyzed by glycerol 3-phosphate dehydrogenase. So in this case, the electrons in here, over here, can be passed on to the mitochondria via this complex or this enzyme called mitochondrial glycerol 3-phosphate dehydrogenase. So it's the same enzyme, it's just another isoform that is being bound to the inner membrane, the inner mitochondrial membrane. Alright? So this one that is over here, right, is in the cytosol. This one is over here is on the mitochondrial membrane. So this mitochondrial version of the dehydrogenase will pass on the electrons that is being found here, all right, via FAD and FADH2. Right, and from here, the FADH2 will enter the electron transport chain, as we have mentioned, to CoQ, coenzyme Q, complex 3, etc., etc. Right, because well, stoichiometrically, experimentally, they have found that if a pair of electrons are being passed via FADH2, 1.5 ATP molecules will be produced. Right? It's quite an odd number, but because there's really no way to count number of molecules, they use chemical reaction to determine the ratio, and it's been found that if you pass it on to FADH2, right? If the electrons are taken up by FADH2, at the end of the whole process, right, through the whole oxidative coupling process, 1.5 ATP will be produced. So in this case, if during glycolysis the glycerol 3-phosphate shuttle is being used, you will notice that the NADH2 is NADH is regenerated, electrons is passed to FADH2, and then pass on. All right, okay, so and then it will go to make oxygen to become water. So this is how we derive 1.5 ATP. And the other shuttle that is being used is a little bit more complicated. And do you notice that in this case there is no pore or channel? There's no transporter. It is just the enzyme dehydrogenase that is being immobilized here. All right, no opening or gate, okay, for things to pass through. But for the second shuttle, you have a malate aspartate shuttle over here. Alright, this shuttle itself is a physical gate, it's a physical channel, physical pore as compared to the previous shuttle. Why? Because over here, instead of using dehydrogenase, the glycerol 3-phosphate dehydrogenase, NADH can also be regenerated by passing the electrons to our oxaloacetate. Right? There is oxaloacetate in the cytosol, and oxaloacetate cannot go through the mitochondrial membrane. It has to change itself, okay? Disguise itself as a malate molecule. So if you see oxaloacetate, the structure is with 2 carboxylic group at the end, right? And the malate version is just the hydroxy version, okay? This is a CO, this is a COH. Alright, so did you see it has taken up the proton, right? It has taken up the hydrogen it's taken up the hydrogen from the NADH, from CO to COH. So only when it is in this malate form can the malate then pass through the mitochondrial membrane via a pore, a transporter, a gate called the malate alpha ketoglutarate transporter. Right? Without the transporter, it cannot diffuse across this really, really tight membrane. So once the gate opens, malate enters into the matrix. This is the mitochondrial matrix. Then it undergo offloading, okay, of the hydrogen back to NAD inside the matrix. And this NADH then goes through the respiratory chain to produce ATP. Right? So complicated, right? I mean, in a case, because of this complicated tight barrier, you really have to transform your materials back and forth. So in this case, okay, NADH, hydrogen is carried by a malate, and then malate gives it up, okay, to NAD inside, two different pools, inside the mitochondria matrix. And then this goes through respiratory chain. So now malate becomes oxaloacetate. The same transformation, but again, the same problem. Oxaloacetate, there's no transporter for oxaloacetate, right? Okay, so oxaloacetate have to change itself again. Okay, but this time it changes itself to an aspartate to get out, right? The direction changes. So in this case, the malate aspartate describes this. So the malate, this transporter describes the entry. The aspartate shuttle describes the exit. Right? It changes itself to aspartate, and let's look at how similar the character is. So now no change on the side. So instead of a CO, it becomes a CNH3. Okay? So over here it becomes OH, over here it becomes NH3. Now as an aspartate, which is an amino acid, then it can go out into the cytosol. Alright, so such a complicated effort just to pass on the protons. And this goes cycles like that. Now, if you notice, then over here, the electron carrier that is being used is NAD, not FAD. So for FAD it's 1.5, but for NAD it's been found the ratio is 2.5. So for every NADH, for every molecule of NADH being passed on down the chain, 2.5 ATP molecules is being formed. Why do we need to know that? Because the calculation of ATP molecules becomes, again, complex when they are being passed on by different carriers. Fortunately for us, there are only 2 carriers. So if we now consider the whole process, now we can see everything in the whole full picture now, right? Glycolysis, citric acid cycle. And let us count the final ATP that is being produced from glucose. Right? So glycolysis, now we have, if you go back to revise, you have 2 NADH, cytosolic 2 NADH from the reaction, and we produce 2 ATP. This is the net, right? Actually 4 is being produced, but 2 is being consumed. So the net is 2 ATP. Right, so if we count the final ATP, now NADH will undergo oxidative phosphorylation depending on the shuttle that it uses, right? That's why we have 3 or 5. If it uses the glycerol 3-phosphate shuttle, then it's FADH, right? It passes on to FADH, therefore 1.5 times 2 will become 3 ATP. But if it uses malate aspartate shuttle, it passes on to NAD, then it becomes 2.5 times 2, which is equals to 5. Okay, that's why you have 3 or 5, depending on which shuttle system it uses. Okay, it depends on which shuttle system it transfers to the mitochondria. And for citric acid cycle, it is quite straightforward. Before it enters the citric acid cycle, pyruvate is being oxidized to acetyl-CoA. 2 NADH is being produced, and this is entirely in the mitochondrial matrix. So 2 times 2.5 will give you 5. And after it goes through the whole cycle, you will have 3 NADH per pyruvate, right? But glucose will produce 2, so therefore 3 times 2 is 6 NADH. And then there's only 1 FADH, okay? But because there are 2 molecules of pyruvate, so you have 2 FADH. And you have also ATP or GTP in this case. This is the substrate level phosphorylation occurring during the cycle. So 6 times 2.5, you get 15. 2 times 1.5, you get 3. And 2 ATP is 2 ATP. So altogether in the total, for each glucose that has gone through glycolysis all the way to citric acid cycle, complete oxidation, you will get 30 or 32 final ATP, or 1 molecule of glucose. Okay, so that's how we derive. Alright, this is the magic number. Right, 30 or 32. Okay? So the pathways are not easy to remember, so you really need to prepare early for the exams. And specifically for glycolysis and TCA cycle, you need to know the structures. So these numbers can get quite confusing. Alright? Okay? Okay? So this is the summary of the TCA cycle. Idea is that it's completely oxidized to carbon dioxide and oxygen to give us ATP. And this ATP will be used, okay, for cellular functions. Just let you watch this video again, yeah? And then get the fatty acid proteins.
It's like reduction rate. It's like
ATP synthase is a molecular machine that works like a turbine to convert the energy stored in a proton gradient into chemical energy stored in the bond energy of ATP. The flow of protons down their electrochemical gradient drives a rotor that lies in the membrane. It is thought that protons flow through an entry open to one side of the membrane and bind to rotor subunits. Only protonated subunits can then rotate into the membrane, away from the static channel assembly. Once the protonated subunits have completed an almost full circle and have returned to the static subunits, an exit channel allows them to leave to the other side of the membrane. In this way, the energy stored in the proton gradient is converted into mechanical rotational energy. The rotational energy is transmitted via a shaft attached to the rotor that penetrates deep into the center of the characteristic lollipop head, the F1 ATPase, which catalyzes the formation of ATP. The F1 ATPase portion of ATP synthase has been crystallized. Its molecular structure shows that the position of the central shaft influences the conformation and arrangement of the surrounding subunits. It is these changes that drive the synthesis of ATP from ADP. In this animated model, different conformational states are lined up as a temporal sequence as they would occur during rotation of the central shaft. Like any enzyme, ATP synthase can work in either direction. If the concentration of ATP is high and the proton gradient low, ATP synthase will run in reverse, hydrolyzing ATP as it pumps protons across the membrane. the rotation of the central shaft, a short fluorescent actin filament was experimentally attached to it. Single filaments attached to single F1 ATPases can be visualized in the microscope. When ATP is added, the filament starts spinning, directly demonstrating the mechanical properties of this remarkable molecular machine.
It's the ATPase story. The ATPase story, do you think it's worth a Nobel Prize? It's a Nobel Prize, ATPase, discovery of synthesis of ATP, and the coupled with the ATPase, this mechanisms have been worth the Nobel Prize in 1997. Right? You know, this year the Nobel Prize was given to a pharmacist, a Chinese pharmacist. Did you notice? Did you hear about that? Yeah, for the discovery of for discovery of artemisinin for malaria. It's a Chinese pharmacist who did not even have a PhD. So, you see, when it comes to research, curiosity is the most important thing, right? And I think it's being shared with a Japanese scientist who discovered a drug for river blindness. And that was found in the soil. Apparently, a friend of a collaborator of mine who knew this Japanese scientist said that he found it because he was golfing one day and then decided to sample the grass sample in the golf course. And that is now, you know how we find new antibiotics, right? In UK, in fact all over the world, if you have a garden in your house, you can send your soil samples to a laboratory in Imperial College. Yeah, then they will look for bacteria in your soil samples and then look for potential antibiotics that are available in the soil samples. Yeah, so you can take a look at that. Right, so we have gone through glucose metabolism mainly, and we said it is the main source of fuel. That's why we need to eat every day to continue to supply glucose to our body. Have you heard of the Atkins diet? Have you heard of the Atkins diet? What does the Atkins diet say?
It means that it's inside.
What does it advocate? What do you eat? Yeah, it's a low carb diet, right? Anybody tried that before?
Then you transport.
Anybody tried that? No, nobody. Anyway, you're so healthy looking, I that. So Atkins diet is what? They ask you to eat low carb, right? But then you just studied glycolysis and TCA cycle. Is it possible? Do you think it's enough? Do you think it's sufficient to power your energy? Power your energy supply? I mean, to give you your 3,000 or 2,000 calories a day. Do you think it's possible? Right? I have friends who are on the Atkins diet and really say that it works and champion it. Okay? So it's partly rational. Why is it partly rational? Why is it partly rational? Okay? Because of this thing called beta oxidation. Right? Okay? So beta oxidation is oxidation of your fatty acids, of your fat stores, right? To provide for energy instead of your glucose. So when you have a low carbs diet, this is what you're forcing your body to do. You're forcing your body to break down your fatty acids to give you through this process called beta oxidation. And this beta oxidation will produce acetyl-CoA. And acetyl-CoA will then go into the citric acid cycle to make your ATP at the end of the whole process that you have seen. Okay, so today we're going to look at the beta oxidation. And fortunately, because the fatty acid structure is also complicated, you don't really need to memorize any structure. You just need to understand the concept and the steps it's not quite like glucose, yeah? This one, you can't memorize structure. You just have to understand how the bonds are being broken, and then and guess, or not guess, but derive how we can get acetyl-CoA out of the fatty acids. So this is the energy content of the food substituents. Right, in your fat diet, what you have, in the fat diet, there are many different kinds of fatty acids, right? But the main component that is, the main component, about 90% of all the lipids that you have ingested, all your char kway teow, your Hokkien mee, what fatty stuff do you eat? Yeah, fried, old chunky, yes, all your fried stuff, right? The triacyl glyceride constitute 90% of those lipids. And of course now we go towards the healthy polyunsaturated fats, right? All your soy oil and all those canola oil, sunflower oil, okay? Those will give you, that sunflower, yeah. This will give you your healthier fatty acids. So look at the energy content in these food constituents. Do you see fats gives you like 37 kg per gram dry weight, compared to your carbohydrate, which will give you a glucose, right? 16. Did you notice the difference? Per gram of the material, you can derive twice the amount of energy, okay, from fats than carbohydrates. Okay, so theoretically, well, actually, most of the cases, you have 30% of your energy coming from breakdown of fats, right? And in industrialized country like us, maybe more, slightly more, 40%, right? So you can see that this is a very good way. And in certain organs, right? In certain organs like the heart and the liver, right? The acetyl-CoA doesn't come from glucose. The acetyl-CoA comes from fatty acid oxidation. Quite strange, right? Heart and the liver. Okay, the heart and the liver. The energy needs come from fatty acid oxidation. Therefore, the glucose part of it is really not the major. I mean, it's not the only thing that is being broken down to give you energy. Fats help out. Okay. Right. So fats, on the other hand, is also very good as a storage. So we said that glucose can be stored as glycogen. Glycogen is incrementally better than glucose in many ways, right? It doesn't change osmolality, and then you can chain it up and just break it down to glucose when you need it. But fats is even better, right? Fats is even better because of this energy difference. And the other good thing is fats. What happens to fats? The solubility is not water soluble. So where is it stored? Where is it stored? Where is the fat stored? The glycogen is stored as granules in the cytosol, right, in the cells. So where are your fats stored? Your adipocytes, your fat cells. Alright, okay, adipocytes, your fat cells. And this fat cells, okay, they are stored as lipid droplets because it's not water soluble. Right, so it's stored as lipid droplets. When it's stored as lipid droplets, it doesn't change again the osmolality of the cytosol. So good, you can store a lot of lipid droplets, right, without affecting the shape, whether the cells will burst or whether the cells will shrink because of changes in osmolality. And the other thing is the fats is very chemically inert. It will not react with any other components of your body. All right, okay? So I think after today's lecture, everybody's going to eat fats already. Yeah. But okay, so all these properties of fats is good, it's lipid soluble, it has more energy, it can, you can store more of your energy in that form in lipid droplets. But it also presents a problem, right? It also presents a problem because the fats, all the other environment in the body is more water, aqueous environment. So the fats cannot be easily transported unless you have the help of what we call bile salts. Alright, so this is your triglyceride structure. Right? Okay, this is the triglyceride structure with CO, and this is the R group. Okay, COO, and then the CH2 are linked up. Okay, triglyceride. Okay. So the bile salts, when you eat your fats, right? Okay, your char kway teow and everything, then it goes into your stomach. The bile salts will have to be released by the gallbladder to emulsify the fats, right? So to form mixed micelles. What do we mean by that? Because the bile salts are what we call amphiphilic. It has hydrophilic and hydrophobic property. So it can orientate itself to hide the fats in the core with the hydrophilic portion on the outside, so that it remains soluble. Okay, it remains soluble in the small intestines. And then the intestinal lipases will then degrade the triacylglycerol and to give fatty acids. Okay, so free fatty acids is the breakdown product. How do we get free fatty acids? So you remove the this part here. Okay, COOH. Yes, COOH. Then you become free fatty acids and other breakdown products. Right, and then it has to be transported through the mucosa membrane to the bloodstream, right? This is only the intestinal tract. It has to go out of the mucosa into the bloodstream. And in this case, now, because other breakdown products cannot pass through the membrane easily, it has then to be converted back to triacylglycerol. And in this case now, instead of bile salts, you have the help of what we call phospholipids, cholesterol, apolipoproteins, again to make it like a micelle structure. This is called chylomicron. Right? This again will help it remain soluble so that it can be transported into the bloodstream or the lymphatic system. Right? Only when it appears in the bloodstream or lymphatic system, then you can utilize it with the help of this enzyme called lipoprotein lipase. Right? When lipoprotein lipase breaks it down to fatty acids, then it can enter the cells. Remember, the cells are where the metabolism will occur, in the mitochondria or in the cytosol. In this case, beta oxidation is in the mitochondria again. Right? We saw the compartment, right? The matrix contains beta oxidation enzymes. So this convoluted way is to transport the fatty acids into the cells for oxidation. Okay, all right. So let us look at okay, this is just a summary of what happens. So you have your triacylglyceride in the free fatty acids, right? And this is the storage form of energy for most organisms, right? The adipocytes. And again, the carbon chains will give you a lot of per gram basis. We saw in the first page. Per gram basis, it will give you much more energy than carbohydrate, about twice per gram. Okay, twice per gram. And the triacylglycerol will be acted upon by hormone-sensitive lipase, your enzymes, that will break it down to free fatty acids to release it into albumin. Right? In order to move around the blood, it has to bind to albumin, a protein. On its own, it has very low solubility, can't move around, so it needs to bind to albumin. Okay, so this is what happens. Now you have your adipocyte, right? Your adipocyte, which is your fat cells, myocyte, which is your skeletal muscles, right? The energy, right, usually is required in your muscles, right? That's why on the muscle side you see beta oxidation, citric acid cycle, respiratory chain to give you ATP. Alright, so now all the fat cells is being stored in adipocytes, different tissues, different cells. How do we mobilize them? They're not in our muscles, right? If they're in our muscles, we'll be in trouble. Right? So in the adipocyte, how do we mobilize them? So they are stored in the adipocytes as lipid droplet, as I mentioned. Right? Lipid droplet, triacylglycerol, the structure that I have shown you. Right, all linked up together over here by the carbon backbone. So now you need some signals. When the glucose level is low, the hormone glucagon, right, will be stimulated. Okay, so this glucagon, this hormone glucagon here, will then, okay, so low glucose will trigger glucagon release, and glucagon will bind to the receptor. Right? Because this is low glucose. It's a signal to tell the body to give me energy. I don't have glucose. Okay, I'm in a lecture hall. I can't go out to buy my bao. Okay, char siu bao. So, okay, so now activate the glucagon receptor and then binds. This receptor binding will stimulate the enzyme adenylyl cyclase to break down or to combine ATP to make ATP become cyclic AMP. This is what we call a second messenger. It's just changes in character a little bit. It's a cyclic adenosine monophosphate. This cyclic AMP will then activate a phosphokinase A, right? An enzyme that has dual function. Number one, phosphokinase A will phosphorylate lipase. Okay, two functions. So number one, protein kinase A, PKA, will phosphorylate the lipase, right, which is enzymes required for breakdown of triacylglycerol. At the same time, it will phosphorylate this protein called perilipin. Perilipin is a protein molecule found on the surface of the droplet. Found on the surface of the lipid droplet. Right? It needs to be phosphorylated in order for this lipase to gain access to the surface. Right? Because the lipase is water soluble. Right? You can imagine that this one and this one is not kind of like immiscible. Okay? Because this is oily, this is in the aqueous phase. So by phosphorylating perilipin, it can help lipase to approach this lipid droplet easier. Okay? So once lipase is on the surface, right, it doesn't go in because it's immiscible. So lipase goes to the surface, then triacylglycerol can then be brought near to the active site of lipase and be broken down to fatty acids. Okay? So lipase gains access to the surface of the lipid droplet and hydrolyzes triacylglycerol to free fatty acids. Okay? This is the mobilization. Okay, you mobilize the fats in the store. Okay, the fatty acids needs to leave in order to go to the skeletal muscles or other organs for citric acid cycle beta oxidation. So it will enter the bloodstream, okay, diffuse out to the bloodstream and bind to albumin, protein, serum albumin, which is this shape here. Alright, okay? So serum albumin will help it solubilize in the blood. Blood is very aqueous, right? Fatty acids, can you imagine it could be like huge long chain, C16, C18. So it could be very insoluble. So albumin helps it along by carrying it like a boat, right, in the bloodstream. So once it is near the muscles, right, there is a fatty acid transporter that will allow the fatty acid to enter the muscles, and fatty acids then subsequently gets oxidized to carbon dioxide and then releases ATP. Okay, so it needs to be carried, right? So now today we're going to look at this process, this transport across, right? Because it cannot permeate, it has to be some kind of pore has to open to go in to the mitochondria and then to go through beta oxidation. Okay. Alright, so this is a description of what I've just mentioned. No, not yet. I mean, this is what we're going to go through. So, fatty acid activation. Right, this is basically this process here to the mitochondria. Imagine this is the mitochondria of the muscles. Okay, so fatty acid activation. Something that's similar that you have seen before is acetyl-CoA activation. Right, and so over here, fatty acids. Look at the structure over here. Fatty acids, right? COOH. Right, and remember we said that at the end of beta oxidation we want acetyl-CoA, acetyl group. So the acetyl group is from here, CO, right? Acetyl-CoA, CO. So how do we get there? Right? So the fatty acids, this is just to show you triglycerides is broken down to fatty acids and glycerol. Nothing is wasted. The glycerol can be converted to glycerol 3-phosphate, which becomes these products. Look familiar? dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Right? This is the midpoint of glycolysis. Right? This is not wasted, right? 5%, about 5% goes to this, and then it goes to glycolysis in the cytosol. Okay, in the cytosol. But fatty acids need to give us acetyl-CoA to go to the mitochondria. Okay, maximally for complete oxidation. So how do we do that? So we have 3 fatty acids that look like that. They're called fatty acids because they have COOH, which is an acid, and the R1 here is terribly long. C14, C15, C16, C20. And not only that, it could be single bonded, it could be all single bond giving saturated fatty acids. Or you can have double bonds in there. Right? So double bonds, polyunsaturated. Many double bonds in the structure. Okay? So over here then we need to somehow form a thioester bond like acetyl-CoA, the same character, CO, then S, COA, CoA. So you need to form a thioester bond between the carboxyl group of fatty acid and the thio group of CoA. Okay, this thio group of CoA and the carboxyl group of fatty acid. Right? So at the end, this process needs energy. ATP will be cleaved to AMP and pyrophosphate to give us the energy. And this reaction occurs at the is catalyzed by enzymes called acyl-CoA synthetases on the outer mitochondrial membrane. All right, on the outer mitochondrial membrane. Why do I say that it occurs in the cytosol? Because we were not quite clear. Okay, the current state of knowledge, we are not quite clear whether it occurs in the cytosol or the membrane space. This is still a question mark. But you see most of the book will quote that this occurs in the cytosol. This conversion occurs in the cytosol. So what is happening over here, right? We have the, at the end of it, this is what we want. Okay, we said that we want fatty acyl-CoA. So did you see what is the difference between the fatty acyl-CoA and acetyl-CoA? Acetyl-CoA just means that the R is a CH3, it's a methyl group. Fatty acyl-CoA means that this R could be anything, C14, C15, C16, C20. Okay, so this R is a